this. I'll get there eventually. So you don't need me to tell you there are lots of good reasons to go hiking in the woods this time of year. I mean, there are beautiful flowers that are just starting to bloom all over the place. There are delicious ramps and nettles to be picked and eaten out there. But the thing that really drives folks crazy this time of year is the morel. You guys know what a morel is? Yeah. I mean, you may know this. A large part, perhaps too large part of the mushroom subculture fanatically obsesses over morels. I mean, they've got organized hunt clubs. They've got these websites where they post morel sightings. And in the peak season, in May, the forests are coursing, literally, with these fungal soldiers of fortune wandering around the woods. I mean, it's a big deal. But now all of us here in this room, we're just a bunch of geeks, right? You know, I mean, you look no farther than back there at Vipin and Jessica and Mickey wearing the GoPro, right? I mean, that's about as geeky as it gets. And the geek mind pursues atavism armed with data, and R, of course. So we might wonder, how could we model the growth of morel mushrooms in the springtime? A good place to start would be to go on these mushroom hunter clubs and scrape their morel postings, their sighting dates, you know. But what else could we do to think about modeling the growth of morels? We'll have to talk a little bit to answer that question, a little bit of background about fungi. So fungi are these amazing organisms that live in organic substrates like the soil and wood and elsewhere. And the mushrooms that are so delicious that we love to eat, they're actually their reproductive organs. They're fruit, per se. And their reproductive cycles are typically annual, and they're heavily influenced by moisture, temperature, precipitation. One of the most fascinating aspects of fungi is that many of them form a kind of symbiotic relationship with plants and trees. And morels in particular tend to like to live among elm and tulip trees as far as deciduous trees go, and there are some species that also like conifers. So we've got a few variables that are interesting here. There's temperature, moisture, and vegetation. Now all of us, as taxpayers, it turns out, we own this amazing constellation of satellites that are almost continuously measuring those variables: temperature, vegetation index. NASA, can you see that, or is it visible to you guys, the picture? So NASA has the moderate resolution imaging spectrophotometer satellite, or MODIS, that measures vegetation coverage globally by looking at various spectra in the near-infrared, infrared, and visible wavelengths. And of course, there's weather satellites. There's all kinds of very interesting information that's all publicly available. After all, we own it. So it occurs to the geek among us that perhaps we could form some kind of a model. We could take some of these morel hunting clubs' websites, scrape their morel sightings, and maybe combine that with NASA MODIS spectral radiometry data from the satellites, and put it together with some other atmospheric information and ground temperature surface measurements. And a first kind of very crude stab at a model might be something like aggregating sightings, morel sightings by date and rough geographic location, and building a simple linear model of those against these variables that we measure so accurately by satellite. Of course, R is spectacularly well equipped to build models like this. I mean, there's a MODIS package that lets you directly interrogate the NASA satellite data and download it into R, right? Of course there is. And there are these beautiful RGDAL and spatial packages that that enable us to read literally any type of geospatial data set, manipulate it and transform it as we wish. And there's an unbelievably comprehensive set of spatial statistics packages for biodiversity modeling, environmetrics, agricultural econometrics. It's really amazing stuff. A good representative example of that would be the vegan package. And of course, there are just all the usual modeling methods that we use every day and know and love about R: linear models, random forests, the whole 9 yards. It's really wonderful for this kind of thing. Now, I know what you're thinking, right? I think I do. the MODIS data only comes out maybe every 8 days or sometimes every 16 days, depending on the data set you're looking at. So this is really just too crude of a model. I mean, to do it right, we'd have to build a time series forecast to predict the near future. For those of you who know about spatial statistics, we'd need to use the appropriate spatial imputation methods on the data. And indeed, linear models themselves require adaptation for spatial statistical modeling. I mean, after all, Bryan Ripley wrote an entire book about this, right? So this is purely a fanciful idea of building a model like this, nothing more. But fanciful or not, if you build a model and make some predictions, you're going to want to interact with those predictions in a way that you can derive value easily and perhaps even explain what you're doing to non-experts. And finally, that's where this HTML widgets idea comes in. You're wondering when I was going to get to this, right? Finally, that's where HTML widgets come in. And so I'll show you before we talk in more detail about that, let me show you some live examples. So here I am in just an RStudio session, and I'm going to load in some data. What I've done here is I've scraped some data from morelhunters.com for morel sightings over time by rough geographic location last year in 2014, and I put that together with NASA MODIS satellite imagery data and some satellite ground temperature estimates data into a modest-sized data set of 160,000 observations or so, and I'll across time and space. And I'll build a simple linear model that's using, these are count data, they're aggregated by space, so I'm just using a simple Poisson model. Now, it takes a moment on my Chromebook to run this, but it's not too bad, it's finished. And then we'll take our model and apply it to this year's data, MODIS data, and see what we come up with. I mean, we want to make a prediction after all. So unfortunately, the most recent MODIS data I was able to obtain yesterday was from March 6th, so it's a little bit out of date. And then I'm going to load those data in and bound them with a rectangle that covers most of the United States. That's what that little, the bracket notation there is. And then I'll just use the normal predict function to make our prediction. Now, when I was playing around with this, I noticed quite different behavior on the east and west coasts. And I think that has to do with the preponderance of conifers on the west coast and different species that favor conifers versus the more deciduous nature of the forests out east. So I've actually split the data into kind of left half and right half of the United States. And I'm going to use the leaflet application that Winston showed us earlier with just a few lines of our code to make a nice little interactive map. And there you go. With only about 10 lines of our code or so, we have our prediction of Morel distribution for March, anyway, of this year, from the model that the very crude model that we built off of last year's data. And it's really cool, right? I mean, Leaflet lets us explore our data in such a natural way. Once you start using this stuff, you're never going to go back to not using it. I mean, you'll think, how did I do this before? But I can look around, and here we go. Okay, in early March, we only see morels there in Georgia, and they're just peeking up into the Kentucky there, and maybe into West Virginia just a little bit, which as a morel hunter myself, I know that that's accurate, even though this is a very crude model. And then out there out west, boy, it looks a lot more interesting. It makes me really want to go visit Humboldt County, actually, for other reasons too. But this is, it's kind of like a Zillow for foraging, right? It's pretty cool. Okay, but that's cool, Zillow, that's kind of neat. But if you really want to get your bling on, there's a new package called 3JS, which is an R interface to this pretty cool stuff called 3JS. And we can do some really blinged out New York Times style graphics. Again, in just a few, in just a few lines of R code. And there you go. So now we have a global, model that we can zoom in on. Yeah. Check that out. I've always wanted to visit the Amazon basin. There's this crazy mushroom hunter named Larry Evans that goes down there every year, and I want to go down there with him some year. That'd be pretty cool. But all right. Enough intro. Let's go back to my slides and talk about what I'm supposed to be talking about, HTML widgets. What is this thing? What is HTML widgets? It's an R package. It's a middleware R package. You actually won't ever use it directly. That defines an elegant and, importantly, systematic way to integrate web presentation technologies and R. And it was conceived and written by Ramnath Vaidyanathan, who used to be at McGill and now he's down in California, and of course J.J. Allaire from our studio. These guys are really visionary. In the old days, like Ramnath and others had lots of ad hoc ways to integrate cool JavaScript visualizations into R. But he stepped back and said, you know, if we had a nice systematic way to put these things together, then it would be kind of an enabling middleware technology that would allow a lot of people to tie web presentation technologies into R in a very clean way. But why web? Why, you know, I mean, you know, what's the deal with web, right? And the deal is that it's pervasive. I mean, it's, you know, the web is in your telephones, it's on tablets, it's in our computers, it's on this Chromebook I'm running right here. This technology touches everybody in every aspect of their lives today. Because of that pervasiveness, web technologies are by their very nature portable. I mean, they're running on every freaking operating system out there, on every CPU architecture. They're as portable as it gets, which is a good thing if you want to deploy software. And again, because of the important ubiquity of the web, there are herds of talented developers writing all kinds of cool stuff. I mean, it's mind-blowing what's out there. You saw it this morning in Mike Dewar's talk, right? Just really cool stuff out there. The New York Times with Mike Bostock and Chris Wiggins and the whole crew there is doing this amazing graphics every day with D3.js that you all know and love. Ricardo Cabello wrote this beautiful 3D visualization framework called 3JS, which I'll show you some more of a little bit later. He goes by Mr. Doob on Google, by the way, which I think is pretty cool. You know, and so that web technology, it's really great, right? I mean, this stuff is cool. We use it and admire it. But what about us? Like, what about, you know, us regular statisticians and data scientists and foragers, right? What are we supposed to do? Do we have to learn, like, JavaScript and OpenGL and all this stuff to use it? It could be very complicated. And that's exactly where HTML widgets come into play. It's a framework that allows package authors to expose these beautiful web presentation technologies to normal R users like us, so that we can create really rich, fluidly interactive visualizations by just writing them in R, using a simple, natural R syntax. And more importantly, those graphics act pretty much like normal R graphics. It blurs the line between where the web ends and R is beginning, the HTML widgets package. And many of the packages are going to be very fast. The 3JS package, for example, uses WebGL, and it's all accelerated graphics on the graphics card. The R user doesn't know or care about that. They just know that their stuff runs really well. Some other important design aspects of the HTML widgets package is that JJ and Ramnath set the thing up so that these things can be used anywhere. We just saw me running some of these graphics inside of an RStudio session, and it popped up just like a regular R plot. You can use them in standalone R Markdown documents, right, that you could then give to a colleague and they could open in their browser and work with your interactive visualizations without even having R if you wanted to. They'll work from Shiny apps. I'll show you an example of that in a moment. So Jared said they were for Shiny. They're not just for Shiny. These things are much, much broader in application. And you can even use them just from command, you know, for you Luddites like me out there who still type in the command line often, they'll also even work there, although they will pop up a browser for you to see them. It's a huge win-win situation all around, right? We, scientists, gain access to this cutting-edge visualization technology, and we don't have to learn anything new. It's really cool. And the visualization gurus who are writing these libraries that are so amazing, they get to put their libraries in our hands. And you know, all of the people in this room, there's a bunch of smart people here, and we will go on to create really amazing things with these technologies. So it's a great idea. All right, so let me show you a few more less silly examples than the foraging example, because I think just to understand what's going on, the best way is to look at some examples. So I'll show you a Shiny app next. I have a write-up about it, yeah. So in my day job, I do a lot of work with genomic researchers. And there's this thing at the National Institutes of Health called the Cancer Genome Atlas, which is a kind of a curated collection of all kinds of genomic sequencing information for about 30 or so broad categories of cancers. These are actually tumor biopsies from patients that were genomically sequenced. They collect and curate and make all of these data publicly available to researchers like us. If you go back in the literature in genomic analysis, a kind of older technique for mining genomic data was to use frequent item set mining or association rules to see if you could discern any patterns in this mess of genomic data. And I'm going to show you a Shiny app that I built literally in an afternoon that does just that and gives us a pretty neat interactive exploratory data analysis tool for cancer and genome atlas data. So let me run that. The code is a little bit longer because there's some Shiny stuff in there, but it's under 100 lines of code. It's not too bad. So it's computing something now on my Chromebook. There we go. So what we're looking at on your left is a panel of parameters for the association rules mining. So there's a package in R written by Kurt Hornig and Christopher Borgelt and many others that does this. You guys know what association rules are? They look for frequent patterns in data. It's kind of one of the most basic exploratory data analysis techniques you can apply. And I'm visualizing those rules on the right with a network visualization using an HTML widget by Christopher Gandrud and JJ Allaire and me called Network D3. So this is D3.js under the hood, but in R, it's just a simple couple of lines. And we get this really neat interactive display of the rules. So what's going on here is, let me just show you what a rule is. So this node says, so I was using cancer mutation data, which counts if there are mutations in a gene region or not. And so these bizarre combinations of letters and numbers are names of genes, really creative names. So what this rule says is that when the gene ATRX, TP53, and IDH1 frequently occur together with mutations in them, in the data set, then so does this type of cancer tumor, which is a certain type of brain cancer. It's a glial cell carcinoma. And the rules are colored by the strength of the association, the stronger ones in red and the weaker ones down in yellow. So we can, this is an output of frequent item set mining. Now down at the bottom, I'm using in this display the data table, or DT widget that Jared was unsuccessfully trying to show you with the baseball data, but here it's working. And it's a beautiful way to display, it's a really cool way to display data frame information. So here we're looking at kind of a data frame representation of the rules that we're visualizing with a network up above. And it's really cool. You can sort on these columns just like you would in an Excel spreadsheet. I can search if I want to look for just things that have IDH1 in them, for example. I get this kind of very responsive interface that shows only the rules that have that gene in them. And now, you genomicists among us in the audience may scoff at frequent item set mining, but I'm not a genetic, I'm just a math guy, right? What do I know? But I did look up some of these genes in GeneCard, and these rules are not crazy, right? I mean, indeed, we know that IDH1 is implicated in glial carcinomas, in brain cancer. So the rules actually pass at least a sanity test. And of course, one of the downsides of frequent item set mining for those, and association rules mining, for those of you who have used them, is that there are a lot of parameters, right? Which I'm not a fan of, actually. Models with lots of parameters are a little bit icky to work with. But with Shiny, you can kind of make parameters more, at least more tolerable, because we can, sorry? we can change parameters and see how they affect our model pretty much in real time here, right? So I just made a change, it grayed out for a moment. Then we get, a lot more stuff back, much more complicated looking graph. I can increase the support, cut back on that. Support is one of the parameters in item set mining. If I only want longer rules, I can turn that up. I might not get any back. Well, there, I only got one back. There's an A-rules viz package, which is very nice. It produces static visualizations in network form similar to this, but it lacks the interactivity that you can that you get for free when you use Shiny with these new widgets. So here we're looking at two different widgets, which I think are very cool. And I should say there's quite a bit of data sitting behind this. The Cancer Genome Atlas altogether consists of quite a lot of information. They have about 20,000 samples that have been fully sequenced. All right, let's go on to another example, another genomic example. Have you heard of the 1,000 Genomes project? It's a project by the National Institutes of Health to fully sequence as many human genomes as they can. It's a little bit misnamed now because they're up to about 2,500 fully sequenced human genomes at the moment. And they make the data available to everybody. Not the raw data, though. What they do is they, we have a bunch of DNA in our bodies that has about 3 billion base pairs in it. It's this long string of protein combinations, double-stranded. And among those 3 billion base pairs, there are only really about 80 million that vary from one human to the next on average. And those are called variants. So these are base pair sites that are commonly variable among Homo sapiens. There's only 80 million or so of those. So what I did was, and the 1,000 Genomes project supplies this variant data to researchers to analyze. And so what I did is I took just a subset of that along chromosome 20. I picked that one because it's a relatively short chromosome. It has only about 1.5 million variant sites on it. And I downloaded that from the web as a text file, loaded it up into R, and converted it into a sparse matrix of dimensions about 2,500 rows, one for each of the persons, and 1.4 million columns or so, one for each of the variant sites. But the data are incredibly sparse. Among all of those rows and columns, there were only about 250 million non-zero cells. In other words, only about 250 million places where the variation actually occurred. And what I'm calling a variation is a variation that occurs on either allele, right? It could be on either strand of the chromosome. And then I used a particularly fast way to compute the principal components, just the first 3 principal components of this large data set. I'd like to point out that, sometimes, I know since we all love R here, sometimes we hear people complain about speed, and, Jared talked about how you can speed things up, and he's exactly right. Data tables, dplyr. What was your third? RCPP, right? Remember those 3 things, if nothing else, as you go away from here. But R in general is not that bad. Like, I went from downloading the raw data file from the web to loading this, pretty big sparse matrix. This is on my home PC, which is a single processor quad-core machine with 16 gigs of RAM, and computing the principal components all the way out to visualization in under 400 seconds, right? So that is not bad. Trust me, R is not fast. If somebody tells you that, you can tell them I said they're wrong, and you can quote me on that. So let's actually take a look at what that looks like. Where is that one? Here we go. So this Chromebook would have computed this a lot more slowly, so I just saved the output, and I'm loading it. And this is actually pretty cool. So there we have it. This is using, again, the 3JS widget, which gives you a 3-dimensional point cloud or scatter plot, which uses the same syntax as the scatterplot 3D package. So you don't have to learn anything new. You can literally just take your scatterplot 3D code, use the scatterplot 3JS function, and it'll look about the same. one cool difference about using the 3JS package is, first of all, it's integrated now into RStudio, or you can put it in a Shiny app or an R Markdown app, which you couldn't do with these other methods for R. And because it uses WebGL, it's incredibly fast. I mean, I've pushed through, even on Chromebooks, I can make point clouds with hundreds of thousands or even 500,000 points and fluidly spin them around and interrogate the data. It's actually quite nice. So this is just one human chromosome, but I mean, it's pretty, even a very basic clustering method like principal components really shows us there are some serious clusters going on here, right? I mean, there's like 4 really obvious ones, and then there's this kind of smear in the middle that might be a 5th, right? And so indeed, we have phenotypic data from the National Institutes of Health. So we know a little bit. They don't tell us too much because otherwise we'd be able to identify it down to a person. But we do know at least their ethnicity. And they have these things called superpopulations. And so we know for each one of the 2,500 samples we're looking at what ethnicity they are. And I'm just going to color these clusters by these known ethnicities from the data set. And it's really uncanny. I was actually shocked at how good this does. So if we look here, notice there's you can label all of the points too and hover over them and see what they are. This cluster out here are East Asian superpopulations. Down here in yellow, we have African populations, South Asian. The blue guys over here are European, and this kind of big mixture here is American, right? So we are the mutts of the world, at least on chromosome 20. But yeah, just a simple PCA. It's quite remarkable, I think, actually. And the widget lets us really check this stuff out and explore it in a really cool way. There are tons more widgets. I've only showed you a few. Because of this enabling middleware technology that JJ and Ramnath have cooked up, there's all kinds of cool people writing widgets now. I would especially urge you to check out Carl Broman's at the top. If you're interested in doing like heat maps or things like that, it's truly awesome. Joe from RStudio has some pretty I'm surprised Winston didn't show the bubbles, actually, because the bubbles thing is a pretty cool demo. So these are kind of ranked by my preference, actually. And I'll give this to Jared so he can put them up on the slide. And one last thing, I'm out of time, so let me just say, if you have a JavaScript library and you know something about programming in that environment, it's really easy to create new HTML widgets and glue this stuff together. The package gives you a very well-specified framework to do this. You only need to write 3 JavaScript functions. Every single widget uses the same 3 JavaScript functions. And you really only need 1 R function and maybe a couple of boilerplate functions to support Shiny. The communication between R and the web uses JSON. We've recently switched everything over to use JSON Lite, which is an awesome JSON package for R. And if you want to be really lazy, the package gives you a scaffolding function that will just prototype the whole thing for you, and then you just have to fill in the blanks to make your widget. So I urge all of you to check it out. There's a website here, htmlwidgets.org, that has a lot more information about this stuff. I think you'll love it, and you'll have a lot of fun with it. Thanks.